Hello, my name is Dr. Rodriguez. I am a science professor. STEAM is an educational movement that combines science, technology, engineering, art, and mathematics. On the iSTEAM channel, we will introduce STEAM fields as access points for experiential learning, inquiry, and critical thinking. Today's episode:
Centrifuging cellular components of pea seeds. In this laboratory exercise, you will separate and analyze the components of a plant cell by performing the processes of homogenization, filtration, centrifugation, and DNA extraction. Separating and identifying organelles that can be viewed with a compound light microscope, as well as extracting DNA molecules from the nucleus of a ripe banana. Centrifuging, separating cellular components of pea seeds. Centrifugation is a technique used for the separation of particles from a solution according to their size, shape, density, viscosity of the medium and rotor speed. The articles are suspended in a liquid medium and placed in a centrifuge tube. The tube is then placed in a rotor or centrifuge machine and spun at a predefined speed. Remove only one of your tubes from the centrifuge and use it for further examination. You will need the remaining tube for centrifugation at a higher gravitation force. Note, you must replace the tube that you remove with another centrifuge tube that contains exactly 14 milliliters of water. This must be done to keep the centrifuge balanced. A second centrifugation will be done for 10 minutes at a gravitation force of 1300 G. Centrifugation at the higher gravitation force will allow sedimentation of chloroplasts and nuclei from the filtrate. Carry out the following 6 steps in order to examine the sediment produced during the first centrifugation. Use the pipette to remove a sample of sediment from the bottom of the first centrifuge tube. Note, you may need to carefully pour out a little of the liquid in the tube in order to prevent a spill. Place a drop of the sediment on a slide and carefully cover it with a cover slip. Use the scanning objective of the microscope to examine the sediment. Note, it will be difficult to identify structures. Add a drop of iodine solution at one edge of the cover slip and allow it to move under the cover slip by placing a small piece of paper towel against the opposite edge. Use the scanning, low, and high power objectives of the microscope to examine the granules that have reacted with the iodine. Name and describe the color of the granules. What is the chemical composition of granules? Observe the layers of cellular components in the centrifuge tube. The liquid on top is the supernatant and it contains small organelles, structures, including mitochondria. The sediment should have a green layer of chloroplasts on top, a brownish layer of nuclei in the middle, and a white layer of starch grains at the bottom. Perform a tetrazolium test on the supernatant by following the directions for the tetrazolium test. Note, tetrazolium chloride is a white chemical that is often used to detect metabolic activity in mitochondria. A bright pink color appears when tetrazolium is in contact with active mitochondria. During aerobic respiration, hydrogen atoms released from mitochondria react with tetrazolium and result in the formation of a pink substance called triphenylformazine. Directions for tetrazolium test. Label 3 test tubes A, B, and C. Use the 10 milliliters graduated cylinder to measure and pour 5 milliliters of the supernatant that is to be tested into test tube B. Carefully pour the remaining supernatant into the graduated cylinder and discard it. Add sucrose buffer to the sediment until the mixture comes up to the 5 milliliters mark on the centrifuge tube. Use the glass rod to stir the mixture, then pour it into test tube C. Pour 5 milliliters of sucrose buffer into test tube A. This tube is the control tube. Add 5 milliliters of freshly made 1% 2,3,5 triphenyl 2H tetrazolium chloride solution to each of the 3 test tubes and shake all the test tubes to mix the contents thoroughly. Place your 3 test tubes beside each other in the rack in the warm water bath that is maintained at 40 degrees Celsius. Observe the tubes at 10-minute intervals. A bright pink color should appear in tube B within 30 minutes. Record your results, using plus to indicate color change and dash to indicate no color change. Explain the results.
I hope this presentation was insightful. Please subscribe and give us a thumbs up if you enjoy learning about STEAM. Please also share this channel with someone you think can benefit from or will enjoy the presentation of the iSTEAM channel. Hope to see you soon!